The overall goal of this procedure is to assess muscle performance and function in zebrafish using touch-evoked response and swimming assays.
This method can help answer questions in the field of neuromuscular research, such as identifying impaired muscle performance or neurodegenerative defects in zebrafish disease models. The main advantage of this technique is that it provides an automated high-throughput method to assess muscle performance in zebrafish disease models.
To perform the assay, place a petri dish filled with approximately 25 milliliters of embryo medium onto an illuminated temperature-controlled stage set to approximately 28 degrees Celsius. Mount the high-speed camera above the dish. Launch the video recording software and set the capture speed to 1000 frames per second to ensure fast swimming speeds can be captured. Position the embryo in the center of the petri dish, clearly visible in the field of view. Press record and then deliver a mechanosensory stimulus by gently touching the embryo with a blunt needle on top of the head. Stop the recording once the embryo has swum out of the field of view or return to rest.
Repeated testing of the same larvae may lead to habituation or promote muscle weakness in some disease models, resulting in reduced response to the tactile stimulus. Therefore, each embryo should only be tested once.
To quantify the swimming behavior,